So we were talking about different types of extremophiles, organisms adapted to develop their whole life cycle at particular physical and chemical conditions, and we started by talking about hypothermophiles, and we had stopped here at the type of molecular adaptations that these organisms have to live and cope with very high temperatures. And here I'm hypothermophiles, and I was mentioning that before, have attracted in particular the attention of different scientists because there are some models on the origin of life that propose that perhaps life evolved at high temperature, and also because at some point scientists, when observing the distribution of hypothermophilic organisms in early trees of life built on molecular markers, in particular ribosomal RNA genes from a variety of species, realized that organisms that were placed at the base of these branches, in particular in the case of archaea and the case of bacteria, and these branches labeled with in red here correspond to hypothermophilic organisms. Well, as I was saying, these researchers found out that these organisms tended to be placed at the bottom of their respective domains, the archaea and the bacteria. In bacteria, we know two different groups of organisms that can live at higher temperatures higher than 80 degrees. And so consequently, it was very easy to hypothesize that the last common ancestor of these of both bacteria and archaea had been hypothermophiles, and perhaps even the last common universal ancestor or ancestor. And so here you have pictures of these different hypothermophilic organisms within the bacteria and within the archaea. However, this question on the nature of the thermophilic adaptation of the last common ancestor has been a matter of debate. And because when people started to explore more microbial diversity, they realized, and also to do better phylogenetic trees, including other markers, other gene markers, in particular protein coding genes, they realized that in particular in the case of bacteria, neither Aquificales nor Thermotogales, so these two types of organisms, were particularly placed early in bacterial trees. So the basal position of these two lineages was questioned. But then there is again a debate, because in the case of bacteria we observe a kind of radiation of organisms, and it's very difficult to say which comes before another one. And so some research, so this is an open question, and some people, in particular in France, have tried to use another approach, and in particular the GC content of ribosomal RNAs to infer the temperature at which the last common universal ancestor lived, and the temperature at which the last common ancestors of archaea and bacteria lived. And in the case of archaea, it's clearly a hypothermophile. In the case of bacteria, a thermophile, perhaps hypothermophile. But surprisingly, their models, and this is all based on, you model the evolution of our GC content in these molecules, pointed out to an hyperthermophilic ancestor for the two prokaryotic domains, but to a mesophilic ancestor for the common ancestor of the two domains. And this is something difficult to explain in biology, but why not? It might be. So this again reflects that this is an open question, and at the same time other researchers concluded just the opposite, that the last common ancestor was hypothermophilic, by using this time a strategy based on their reconstruction, the theoretical reconstruction of ancestral proteins, taking proteins, and in particular they used this nucleoside bisphosphate kinases, a variety of them, and so they inferred in silico which would be the ancestral sequence here, and then they could synthesize the protein and test the optimal temperature for the activity of the protein in vitro. And they observed that these temperatures were very high, and so that they concluded that most likely the last common ancestor was hypothermophilic. The question is not closed, it is always open, but you know that there is a debate because these people have also used protein sequences, and they again conclude that the last common ancestor was not hypothermophilic, so the debate is open. But that I just wanted to mention that hypothermophiles are important also from this perspective on early evolution and the nature of the last common ancestor. And I will come back onto this question a bit later, but more related to the domains of archaea and bacteria. So we have explored organisms that grow and develop at very high temperatures, and on the opposite side of this gradient, temperature gradient, we have organisms that live at very low temperatures. They are called psychrophiles, and well, you find them in this type of environments. You can't find them in Antarctica. The record of metabolic activity at very low temperature has been registered at the South Pole, going from something between -12 to -17. Some authors argue for -20 degrees. But here in this case, we are at temperatures, especially when you go to these values, -15, -20 degrees, where you detect some metabolic activity, but not necessarily duplication. This means that the whole life cycle is not completed. So you have residual metabolic activities, for instance, to repair DNA and prevent that mutation end up with everything, or for instance, to repair DNA breakages. So you have still some remnant activity, but you cannot really develop at these very low temperatures. So this is something, the lowest temperature at which an organism can complete the life cycle is a bit unclear. Paradoxically, one of the features of this type of very cold environments where you can have ice or water ice is that the water activity is very low. You don't have liquid water, and so in a sense, as we will see later, many of these organisms that live in places like Antarctica have adaptations that mimic or that are very much the same than those of xerophilic organisms living in deserts, even if they are hot deserts, because they are facing this hydric, this water activity limitation. And one of these adaptations consists into living within rocks. So this is a particular rock in Antarctica, and you can have communities. So you see the colors. This is a layer of photosynthetic organisms, and here you have organic matter and likely many heterotrophic microbes. And then they get into the rock for two different reasons, actually. One is that they protect themselves from UV radiation, because in particular in the poles where you have a big ozone hole, so you may have problems with ultraviolet radiation, but also because within the rocks you can have water condensation, and because perhaps you have increased salinity, etc., and then these organisms can live there. So this is one of the potential adaptations that you see in this type of environments. So these are pictures of some organisms that you can find in this type of environments, and these are eukaryotes, because we saw that in the case of high temperature we have only prokaryotes, and at the very upper limit only archaea. And here you can have also, you have bacteria archaea, but also many different types of microbial eukaryotes that can live, for instance, living on snow. When you see this kind of pinky areas, these are eukaryotic microalgae that live there on snow. As in the case of hypothermophiles, the enzymes of these organisms that can perform their activity at very low temperatures are very interesting for industry, for a variety of processes that can take place at very low temperatures. So again, I put here just a table showing that these enzymes are available commercially and that there are many. So this is something that is interesting for industry. And then, as in the case of hypothermophiles, you have that temperature that these organisms also adapt to the potential deleterious effects of low temperature. Low temperatures can lead to protein inactivation, or they can limit, in the case of nucleic acids, the accessibility of proteins to the sequences that they need to target. Also, this is highly limiting for nucleic acid-dependent processes. And the other effect at the level of the membrane this time is just the opposite as in the case of high temperature. Low temperature can increase membrane rigidity and so make membranes too impermeable to a variety of metabolites and ions. And so the adaptations that these organisms have tend to counteract these effects. So in the case of the DNA, the problem is just the opposite. So you have a collection of proteins and strategies to unwind DNA, because DNA tends to be too tightly packed. So you have enzymes such as gyrase, which actually acts in the opposite way to reverse gyrase. It just unwinds, creates negative supercoiling, so it tends to unwind the double helix just to maintain the geometry of DNA in the optimal way for a variety of proteins to interact with it, and make replication, repair, transcription, and so on. In the case of eukaryotes, instead of this protein gyrase, which is a protein that evolved in bacteria, and some archaea also have it, they acquire it by horizontal gene transfer, you have histones and the activity of other topoisomerases that create a little bit this same effect, this unwinding the double helix. At the level of the cytoplasm, you can have antiferricin molecules, many compatible solutes, many small proteins, for instance, or sugars. And then at the level of protein adaptation, what we have is all kinds of adaptations that tend to increase the flexibility of proteins. So like reduced hydrophobic cores, less charged amino acids in the surface, and more lateral chains that all tending to increase this flexibility. And we have also, and this is what is interesting actually for biotechnology, a very high affinity in the case of enzymes for the substrate. Okay, so decreased activation energy. And you also have specific chaperones and cold shock proteins that help to get the proper fold and prevent proteins to be too compact, actually, to fold properly under these conditions. And in the case of membranes, what you observe is a higher insaturation at the level of the lateral chains because this increases flexibility. In many cases, you see branch lipids, which is also has also the same effect, and well, a variety of adapted transporters, etc. So, in summary, when you compare the adaptations to high temperature and to low temperature, not surprisingly, you find like opposite or mirror properties. On one hand, you adapt to introduce positive super turns on DNA and higher resistance to thermodegradation, unwinding on the other side, more compact proteins versus more flexible proteins, lower permeability in the case of hypothermophiles, and higher permeability membranes in the case of low temperature organisms. So having this a kind of adaptations, molecular adaptations and cellular adaptations in mind, it is also interesting, and also the idea of a thermal adaptation during early evolution. We can try to relate the two of them. And actually, this is a bit of a side story, but I hope you find it interesting. We can ask whether horizontal gene transfer could have played a role in the early evolution and in the temperature adaptation of major domains of life, in particular of bacteria and of archaea. Just a brief introductory slide to tell you about horizontal gene transfer for those of you who are not biologists. So in general, adaptation proceeds, and this is how traditionally people, evolutionary biologists, study how organisms adapt in biology. It proceeds by mutation that introduces variation, and then selection can act on the different variants that are produced by mutation. So this is the traditional way. But in recent years, let's say the last 10, 20, 30 years, there is increased evidence that at least in the case of prokaryotes, and possibly it's more general phenomenon, horizontal gene transfer, that means the acquisition of a gene from a distant organism, is a very important process in evolution that can act in a sense like mutation, but you mutate much more and you obtain something new. And so it can have a very important consequences in evolution. It is very widespread in prokaryotes, as I said, bacteria and archaea. It mostly involves metabolic genes, because metabolic genes in general first provide an immediate function and advantage in terms of potentially degrading a substrate or producing such and such molecule. And in these functions then, or many of the enzymes involved in metabolism, interact with a substrate and give a product, but don't have to interact with many more proteins at the same time. So it's easier for them to be changed during evolution without causing deleterious effect in your interacting proteins, for instance. So this is in any case an observation. Many metabolism genes have been transferred among prokaryotes, among bacteria, among archaea. This horizontal gene transfer contributes to innovation and adaptation in many different ways, because you can, for instance, incorporate a new function from a distant organism where the function has evolved via importing the gene coding for the particular protein. But you can also import a protein for which you have already a gene encoding a similar protein, and maybe mutate this protein because it's in a double copy, and then this can lead also to innovations by getting released of selective pressure in a duplicated copy. And of course, horizontal gene transfer, if you pick the right gene, confers an immediate selective advantage. And this is the reason, by the way, why you retain a gene that you import. Otherwise, you lose it. But horizontal gene transfer can have some potential negative effects, and this is why many horizontal gene transfers are not retained. So you can, if you import a gene, you can have a potential fitness cost, because depending on the expression context, imagine you get a gene, but of course each organism has the machinery adapted to a particular codon usage. And if you get a gene from another organism having another codon usage, this gene may not be adapted properly to the genomic environment, to the genomic context, and to the machinery of the cell that is biased towards a particular codon usage. So this can impair the expression of the newly acquired gene. You could also have metabolic incompatibility if you get a gene that performs the opposite function of your favorite function in the cell, and of course you will get rid of this gene. And in the case of multiprotein complexes, if you get one of these genes that encodes a protein being in a larger complex, the interaction between the different components can be affected, and this can impair the function of the multi-protein complex. And there might be other deleterious effects in a particular cell. So this, in these cases, in general, there is a trade-off, and you can only retain a distant horizontal gene transfer gene if the function is really advantageous. It means that the benefit that you gain is higher than the potential limitations of the import of this gene. So I think this seems clear. Okay, so this is a very general introduction on horizontal gene transfer. Now we know, and this is one example of this particular sentence here, that long distance, by long distance I mean the acquisition of a given gene from a very distantly related, phylogenetically distantly related organism. Let's say in an interdomain horizontal gene transfer, a gene that is traveling from bacteria to archaea, for instance, or vice versa, or between two different phyla within bacteria. So one example of this is the adaptation of these bacteria, Aquifex and Thermotoga, so the two groups of hypothermophilic bacteria, to high temperature. So when the genomes of these two organisms, Aquifex and Thermotoga, these two species, were sequenced by the late 1990s, people discovered that there were some genes that were of archaeal origin, and between these genes, for instance, reverse gyrase, which is a marker of hyperthermophily and is a protein that is essential for hyperthermophiles. And so the idea was that these bacteria adapted secondarily to high temperature by acquiring genes from hypothermophilic archaea. So this is what happened. This independently of the nature of the common ancestor, which we don't know. Some people say it was a mesophile, other hypothermophile, but what we know is that the ancestor of archaea is a hypothermophile, and the ancestor of bacteria possibly something between a mesophile and a thermophile, but a moderate thermophile and not a hypothermophile. Okay, now what happens in the case of archaea? What we know for sure is that the last common ancestor, nobody discusses this, the last common ancestor of archaea was for sure a hypothermophile. So here you have a color code indicating the optimal growth temperature. So red means hypothermophile as before, and then the more you get to blue, the more mesophilic or even psychrophilic you are. And so you have that the root of the archaeal tree, which is the traditional root, is placed here. Other people place the root of the archaeal tree here. There is a lot of discussion, but it doesn't affect our view on this because the two places correspond to a hyperthermophilic ancestor. And then you have many different lineages within the archaea that are hypothermophilic today, and these are red, but many that have adapted to lower temperatures. And you will see here that these groups are not necessarily forming a monophyletic group, that is, they are not joined together. You have lineages of hypothermophilic archaea separating groups of mesophilic archaea. So, for instance, these groups, this means that these groups adapted secondarily from a hypothermophilic ancestor to lower temperatures, to mesophily. Okay? What to say about these groups? Some of these are very well known. So you have many methanogens that live in sediments, and also halophilic archaea living in high salt conditions that form a monophyletic group, and they are very diverse. And you can find some hypothermophilic methanogens living at the base of the tree, but this grouping here, they are really mesophilic. And then you have other groups of organisms like this Tomarchaeota and these groups 2 and 3, which are essentially uncultured organisms In the case of Tomarchiota, there are few species that are cultivated, but there are many organisms that are living there in the environment for which we don't have culture members in the lab. This is a highly diverse group that consists essentially of organisms that are chemolitho-autotrophs, gaining energy from the anaerobic, aerobic oxidation of ammonia to nitrite, and then that are autotrophic. And these groups 2 and 3, not a single member in culture, but they are very abundant in oceans, some in deeper waters, but, well, so many lineages. And these groups are not the only mesophilic groups in Archaea. There are many others that we know based on these environmental studies. You may have heard, for instance, about the Lochiarchaeota that belong to this group here, and that seem to be more related to eukaryotes. So now some people think that these Lochiarchaeota are the closest Archaea to the ancestor, or one of the potential ancestors that made a symbiosis to originate eukaryotes. But there are other groups in other places of the tree, and all these groups have adapted secondarily to mesophilic conditions from hypothermophilic ancestors. Now, it happens that now we have genomes from many of these lineages, including for lineages that are environmental, because we can get their genomes from metagenomes, as we saw before. And so when you analyze the phylogenetic affinity of the genes belonging to these different lineages, including thermophilic and mesophilic lineages, well, you realize that many of these genes, especially in groups of mesophiles, are more related to bacterial genes or have a bacterial origin. And actually, there is a very nice correlation between the proportions of genes acquired from bacteria by Archaea in the case of secondarily adapted groups to mesophily than in the other groups. Okay? They also have larger genomes as a consequence. And not only that, not only they have more genes imported from bacteria, but it happens that many of these groups of Archaea have acquired the same gene independently from different groups of bacteria. And this is one such an example. All that is in black here are bacteria, and then these 3 groups of mesophilic Archaea acquire the gene from different bacterial donors. Okay, this is how you should read this tree. It means 3 independent acquisitions of the same gene. So this means that this gene might be important for a function that potentially is related to mesophily. And when you count all these genes that are, so this means 3 independent horizontal gene transfers. And when you count all these genes coming from bacteria and that are shared between these different groups of Archaea that are independent adaptations to mesophily, well, you count quite a number. And these genes are essentially related to energy metabolism, membrane transport and lipid metabolism, and also amino acid transport and metabolism. Some of these functions may be important for the adaptation of these Archaea, whose ancestor was hypothermophilic to mesophily. So that's the hypothesis. So we can really hypothesize, and the data are compatible with that, that in the same way as we think now that these hypothermophilic bacteria adapted secondarily to high temperature via the acquisition by horizontal gene transfer of genes from hypothermophilic Archaea, you can imagine that mesophilic Archaea evolved or adapted secondarily from hypothermophilic ancestors towards mesophily or to mesophily by importing genes from bacteria, and very often the same type of genes. So that's something that more data should validate, but there is quite a solid support for this idea. Okay, so that was for a little bit of early evolution, and now we come back to a more descriptive part of the talk, continuing with our trip by all these extremophilic organisms. Now we are going to halophiles. Halophiles are organisms adapted to live at very high salt, up to saturation. Not surprisingly, the most or the best adapted organisms to high salt concentrations are again archaea. And then you can have very special archaea, such as this haloquadratum. This is the name of the genome. You can imagine why. Because they have really very funny shapes or very unusual for biology. So these are organisms that you find in this type of solar saltans, but also in deep sea brines, for instance. Many of these organisms form the nucleation center of crystals, salt crystals, and the red color here, as here, is due to one pigment in the cell wall of these organisms that is related to a particular protein, one kind of rhodopsin, proteorhodopsin, bacteriorhodopsin, that allows these organisms to get some extra energy from light. So this makes some of these archaea making phototrophs, not photosynthetic. They cannot fix CO2. They can just gain an extra energy. They are photoheterotrophs. They gain some energy from light just by using this particular protein, enhancing the proton gradient across the membrane. Okay, and the protein is called bacteriorhodopsin, but it was isolated first from one archaeon. But when archaea or halophilic archaea were thought to be bacteria, and that's what's funny about this bacteriorhodopsin, which is an archaeal rhodopsin. Okay, so adaptations. Again, as we have specific adaptations to life in high salt conditions. Of course, the major risk here is osmotic catastrophe. So what these organisms do is to increase, well, either to use compatible solutes, but this is something more present in halotolerant than in halophilic, truly halophilic organisms. And the most halophilic organisms, namely the archaea and one particular kind of bacteria, they accumulate potassium in the cytoplasm up to 4 molar, which is a lot. And so there are specific pumps involved in this. And then they use also sodium proton antiporters to get rid of sodium and include protons instead in the cytoplasm. The proteins tend to be negatively charged, and also the surface of the cell, thanks to the presence of many polysaccharides that are charged, exopolysaccharides. And so the thing that the proteins are enriched in negatively charged amino acids can be even seen at the level of metagenomes when you analyze these metagenomes in bulk metagenomes, so community genomes from from environments at different salinity. So let's say from almost seawater, this would be 3.5, 3.8, towards saturation, 37%. And what you see here is that the pI of proteins predicted from the metagenomes decreases progressively with an increase in temperature, so that the pI of proteins predicted from genomes of organisms living at saturation have the lowest pI. So the pI would be the pH at which a particular molecule has not no net charge in this case. So you can even see that in predicted proteins from metagenomes. And as in the case of hypothermophiles that I showed you before, we also observed a decrease of diversity when we go towards where the most extreme limit or the most extreme condition. So here, if you analyze, in this case, diversity from metagenomes along a salinity gradient, we are here at 19% salinity, which is already quite a lot. But if you go to saturation, you see that there are less parts in this pie chart, and that you get a reduction in diversity, with essentially most of the diversity belonging to this Euryarchaeota. This is the group of archaea where the halophiles belong to, and then one particular kind of bacterium. So this in particular correspond to not only the Euryarchaeota or halophiles, but to a particular species, that this is a square archaeum. And also, in the case of bacteria, you have very specific microbe, Salinibacter ruber. You can see the adaptation in the name, which is mimicking the adaptations of archaea in terms of potassium accumulation in the cytoplasm, and possibly also with a lot of horizontal gene transfer between the two organisms. One funny thing is that this Haloarchaeon quadratum, because I told you before, it takes it gets a little bit of energy from light. This explains a bit the shape of the cell. You see here these lighter areas. This corresponds to gas vacuoles, and using these gas vacuoles, the cell controls the orientation of this flat surface to the sun, so that the cell acts like a solar panel. So that's a funny adaptation. And of course, I am talking about a lot about metagenomics, because it is really a tool for discovery, and the use of these metagenomic approaches have revealed the presence of new lineages that appear to be specifically associated to this type of environments. And here is the case of this particular group of organisms, the nano-Haloarchaeota, which correspond to very tiny cells with very relatively small or very small genomes for the average of bacteria. And so you can see these tiny cells compare with the cells of square archaea here in blue, okay, with color with DAPI, which is a stain, a DNA stain. Okay. You have also halophilic, alkaliphilic organisms. So we move to pH. Why I'm jumping? Okay. Alkaliphilic organisms, many of which are actually halophilic, like those living in soda lakes, which are saturated in salt, but also have very high pH, and many lakes in Africa have this type of feature. So you have organisms that are at the same time halophilic and alkaliphilic, because they adapt at high pH. But you have also other lakes that are not necessarily salt-rich, like the Lake Van in Turkey, where you have this type of structures, stromatolitic-like structures, and which has a pH of almost 10. And there are many lakes, or like the Alchichica Lake. This is a lake that we have been studying for several years already, which contains also stromatolites. So it appears that many of these stromatolitic or microbialites, this is a general term, are frequently found in this type of alkaline lakes. And we have been studying these organisms, and even discovering organisms like this Gliomargarita lithophora that have particular properties. They can produce carbonate precipitates of calcium, magnesium, this is magnesium, strontium and barium, which is something quite atypical. So these organisms come from this lake. So many stromatolitic formations in this type of alkaline lakes. And you have, on the opposite side of the gradient, organisms that live at very low pH. We call them acidophiles. These are very often places that are enriched in many metals, because the two features coexist in nature, in particular in mines, in metal mines, iron mines, or many other types of metals, arsenic, etc. The record of an organism growing at low pH is again belongs to an archaeon, Picrophilus oshimae, which was isolated from volcanic region, so it's a thermoacidophilic organism living at 60 degrees more or less of optimal temperature and at pH 0.7 as optimum, but it can grow at a negative pH, or at much higher proton concentration. And many of these organisms actually are used for biolithium, for instance, because they can enhance or catalyze or they can remove or leach metals from minerals. Many of these are found in places where you have pyrite, for instance, that is naturally oxidized to sulfuric acid. This, of course, decreases the pH, but you have organisms that increase this process by a lot. One of these organisms that is very famous because it's really one of the best adapted organisms to very low pH is Aciditiobacillus ferrooxidans. This is a bacterium that is highly flexible or versatile metabolically, so it can oxidize iron and get energy in this way, but under aerobic conditions, it means with oxygen. But in cases where it grows anaerobically, if there is no oxygen, it can also gain energy by reducing iron 3 to iron 2 using reduced sulfur compounds. So it's an organism that can gain energy in any possible way in this type of settings. And it is not only using iron, but it can use the whole variety of other metals. And so this is one example that is, and there are many others that are related to these organisms that are interesting, in particular for mining operations, because you can leach these metals from minerals thanks to their activity. But in low pHs you have also a variety of eukaryotes and not only prokaryotes, and this is these are pictures from the different eukaryotic organisms, some fungi, some diatoms, or these are euglena, so algae, some heliozoans, and you have here different flagellates. And all these organisms are coming from the Rio Tinto in Spain, pH around 2.5 on average along the river. And there is a wide variety of eukaryotes in this type of environment. Okay. So you can study these organisms using metagenomics, for instance, and this has been done, and this was among the first studies that were done on metagenomics to try to reconstruct genomes from metagenomes. And so in one of Pioneer's study, these people, Tyson and co-workers, reconstructed the genomes of two different organisms, one archaeon and one bacterium, living in acid mine, in acid mine drainage at pH around 1. And you can, from these genomes, reconstruct or predict the theoretical metabolic abilities of the cell just by cartooning the different proteins or annotating the proteins and predicting the functions. So in these cases, you have organisms that oxidize iron in this type of environments. And this type of analysis have also revealed new groups of organisms that this time are surprising and were surprising at the time, 2006, because these Arman groups, which were specifically detected first by metagenomic sequencing, were then subsequently observed by fish, fishing them with a probe. And these organisms are particularly small. So you have here size, it's 200 nanometers. So these corresponded at the time at the cells that were at the smallest possible limit thought at the time for life. For life, or relatively complex life, it means true life with a metabolism. And so these have particular shapes sometimes, so the cells are relatively flexible. So these are these Arman archaea. You can develop biotechnological application, and many of these organisms acidophiles in particular, but also other types of extremophiles, are being used to try to develop microbial electrochemical systems. So in a sense because you can exploit the capacity of a microorganism to perform particular redox reactions from a substrate to a product, and then liberating electrons that can be passed on to an anode, or organisms that can take electrons from a cathode. So you can have this type of organisms that actually enhance or catalyze activities that lead to electrochemical systems. So people are working on this system to try to develop batteries or biobatteries in a sense. I haven't mentioned the adaptation of acidophilic organisms, but I forgot this. But one thing that these type of organisms do is that even if they are adapted to live in very low pH environments, these organisms keep an internal pH that is much higher. So one of the adaptations is to get rid of protons. Okay, so the most acidophilic organisms, I mentioned before Picrophilus hoshimae, living at pH around 0 optimally, it has an internal pH of around 4.5, which is quite high. And most other acidophilic organisms have a quite neutral pH between 6 and 7. So the adaptations of these organisms are largely passed by maintaining an internal pH that is compatible with most biochemical reactions. Okay, so we go now to barophiles or piezophiles, organisms adapted to very high pressure. So where do you find these organisms? You find them in at the bottom of the oceans at very high pressure, or even below the oceans. So most organisms, or planktonic organisms, and even in the sediments in oceans, are barophilic, or barotolerant at least, or piezotolerant, because they live at large surface of the planet is covered by oceans, but the average depth of the oceans is something between 3,000 and 4,000 meters. So these are it means that the most organisms in these environments are really adapted to these low pressures. And then, well, the deep sea is also extreme for a variety of things. There's no light, but there are highly oligotrophic conditions, so this limitation in nutrients, the high pressures, and also low temperatures. So you have organisms that develop there, and they are particularly adapted to that combination of parameters. You have also xerophiles, organisms living in deserts at very low water activity, and many of these organisms are developed in the form of biofilms that cover rocks, or even within the rocks, as I'll show you a picture just in the next slide. And so some of these biofilms are called desert varnish because they are dark like this, and then these organisms are essentially bacteria, growing very, very slowly and highly pigmented to protect themselves from ultraviolet radiation. And actually, I'm going to talk now about radiotolerant organisms because, well, one of the famous organisms that are tolerate, or the record is Deinococcus radiodurans, that is or can cope with significant levels of ionizing radiation. And actually, this organism is essentially a xerophilic organism. So it's an organism that is a bacterium, and it thrives essentially in desert areas, or in biofilms colonizing rocks, in places where desiccation is important, and where you have UV radiation. So Deinococcus is very well adapted. In general, these organisms are to avoid the damaging effects of ultraviolet radiation. In particular, Deinococcus, in general, these organisms are pigmented, and in particular, Deinococcus has extremely has an extremely powerful machinery of DNA repair. So even if ultraviolet radiation cuts the DNA in many pieces, and you can do experiments with ionizing radiation as well, this organism is able to repair the genome very rapidly. Within a few hours, you can recompose the genome. And it is so because, as I said, it has a very powerful repair machinery, but also because it has several copies of the genome, so that you can better recompose your genome or recombine to repair. Okay, so that explains why it is also adapted to ionizing radiation. It's not because nuclear plants were there at the beginning of life, but it is because it adapted to desert conditions and to ultraviolet radiation. And many of these organisms are living, as in the case of Antarctica, within rocks, because you can protect yourself better from ultraviolet radiation, and also because you can get water condensation better. And that's exactly what happens in one of the most extreme environments you can think of. From this perspective, there is some alite rocks in the Atacama Desert, where you have practically no rain, and in some places there is no historical record for rain. And but you have organisms living without these halite blocks. And if you place probes for 1 year or several years within these different depths within these rocks, you can realize that even if these organisms effectively live within the rock, and they live at places where they still have light, because many of these organisms are cyanobacteria, and they are accompanied by many heterotrophic bacteria that depend on the primary production of these organisms. But also in regions where you can have periods of water condensation along the year within the halite, in the absence of any condensation outside. Okay, so even if you have no liquid water whatsoever outside the rock, you can have at some particular events along a year between 3,000 and 5,000 events of water condensation, and these organisms wait until they have these conditions to develop and carry out their life cycle. Another environment that shows this type of combination of very extreme conditions is the deep subsurface, which is maybe interesting in the case of astrobiology, but also here in our planet, where organisms live. So in this case, organisms live in the continental or oceanic crust, so at high pressure. You can find them in deep mines, in deep aquifers, in these systems, or in caves, where organisms live in the fractures and crevices of these minerals, rocks, and potentially using a variety of chemolithotrophic metabolisms, but perhaps not only that. So how to access these microorganisms? One possible way, and there is a consistent number of studies on this area, is by drilling. So here is an example of a study derived from the International Ocean Drilling Program, which has been drilling in many places around the world. And that shows that in different, so this is a graph showing the subsea floor depth. In this case, is sediments, but it could be rocks. So you have a decrease, of course, of total microbial. In this case, the marker used is the presence of polar lipids. And there is a decrease, showing that with depth you have less and less microbes. So there is a mixture of active microbes, but also a decay, yeah? So a kind of mineralization with time. So we are entering the fossil record and the geological record in a sense. But along this gradient, so you see this trend, you also see that the number of archaeal lipids increases. So suggesting, because these are intact lipids, that you have still a quite significant activity of archaea in particular, perhaps related with the fact that maybe you have higher temperatures at deeper areas, and then that there is an activity and there is not only decay. And this you can test it in other type of environments. For instance, this is a study that we made in a deep mine, so at around 760 meters. So this famous cave of crystals, where you have giant gypsum crystals in Nica in Mexico, is here, much or more at the surface, 300 meters depth. And here you can have access at the aquifer. There is at this level, more or less 700 meters depth, and then you can avoid surface contamination. And it happens that in this aquifer you can identify the presence of archaea, and in particular of particular groups, tomarchaeota, that are ammonium oxidizing and that are thermophilic. And this particular candidate division OP3 in bacteria that are potentially living in this aquifer at an extreme nutrient limitation. So how do these organisms do? This is one particular and very extreme condition, low energy environments. And actually there are some other studies of microorganisms living underground at more or less deep locations in underground water. And these organisms tend to be diverse, but not as diverse as in surface waters, but they tend to belong to particular groups of archaea and bacteria. And many of these groups are extremely small in size, to the point that they pass the filters that are used to retain most bacteria, which have in diameter 0.2 microns. So these organisms are really at the very, very limit of what is needed for a cell to survive, being independent or more or less independent. And so the size here, the scale bar is just 100 nanometers, so you have space for a tightly compact genome, a few ribosomes between 40 and 50, and not much else actually. So, but these organisms seem to make a living out of it, and they seem to also harbor viruses that can affect them. These are bacteriophages attacking them. And we don't know much of these organisms, so this is past year, but they exist. So all these deep sea and deep subsurface microbiota has raised a variety of ideas, and in particular the possibility that some of these organisms can really be independent or fully independent of light, which is and that they may perhaps depend on hydrogen produced geochemically or geologically. Okay, because you need for life to go on in this earth. On this earth, you need a reductive power that it is gained essentially by photosynthesis, fermentation, or CO oxidation, and many of these processes take place thanks to photosynthesis, which is the major dominant process in surface. But it might be that some other processes lead to hydrogen that can use as a reductant in this type of deep sea and deep underground systems. Open question. So, as a summary, if we summarize the range or the intervals of different physical parameters or physicochemical parameters within which life can exist, we can see the temperature more or less between -20, let's say something, to plus 120, pH from 0 and even negative pH to, I put here 12 because we don't know many places on Earth at higher pH than that, and so if there are no particular environments at that pH, you don't have the organisms that are adapted to them, but perhaps they exist somewhere on Earth, I don't know. Salinity, you need some salt, but we don't know which is the lowest limit for life from that perspective, because not much interest actually. But you can live up to saturation, or you find organisms living at under saturating conditions of a variety of salts. Pressure, we mentioned this before. We have no idea about what's the minimal pressure for a complete cycle to take place, but we know that you can go to very high pressures under the deep surface, for instance, or at the bottom of the oceans. But as we evoked before, this is conditions where organisms carry out the full life cycle, that they are adapted to live at these conditions and they can reproduce under these conditions. But many organisms can undergo and bear conditions that are far more extreme than these ones, just by entering dormancy, or producing resistance forms that are a way of dormancy. So you have here a variety of spores. So this means that you can stay for some time. It can be months, years, or thousands of years in some cases, in a dormant state, and that can lead you to mortality if conditions are not recovered. But if the conditions are right again after, let's say, several hundred years, you can again, in a sense, resuscitate your cell and live again, or perform the full cycle. And actually, many of the natural or some of the two of the best ways that we use in the labs to conserve cells, cryopreservation and lyophilization, do exist in nature already. For instance, in permafrost, or you can freeze cells. We microbiologists, we usually have, if you culture organisms, you can freeze them at -80 degrees or in liquid nitrogen at much lower temperatures, and you can keep your cells there for many years, or sperm cells, whatever, and to assist human reproduction. You use this kind of temperatures, and then you recover the organisms. Nature does the same in the permafrost and other polar, very cold areas. And you have also lyophilization. You desiccate completely your cells, and these are tubes of cells that you can buy from a culture collection, and then they can send you the tube of lyophilized cells. You just add some liquid medium, and cells can grow again. And this is exactly what happens with some archaea, for instance, that live within crystals under complete desiccation. You add some culture medium, and they grow again. Okay, and this, of course, we were mentioning these exposure experiments, where you can take some of these organisms and expose them to outer space, and they can at least endure some periods of radiation out there, and then some of them grow after some time because they have resisted but not lived under such conditions. And of course, this will be more or less my final comment about these organisms. These adaptations and these properties of extremophiles have relevance for astrobiology in general, because they extend the limits within which we can hope to find life eventually in other planetary bodies or other satellites or whatever. So this is important. We learned from exploring microbial diversity on Earth that the limits to life are not necessarily restricted to fresh water and oceans as we know them, but also to other much more extreme conditions. And there are many places where to look for. I'm not sure that life exists in our solar system, but when it is a possibility. Some people are thinking on Europa, for instance, and in particular, this could be a possibility if there is a liquid ocean under the crust, which seems to be the case, and you have enough nutrients in this water space. Perhaps if there is some hydrothermal activity, so why not? and particular interest or analogous systems to study on Earth are permafrost and very cold environments. There is also Mars, of course, and not because life exists today on the surface, it is likely not the case, but because water existed certainly in the past for some time, and perhaps it is not impossible to think that at some point life could have evolved in Mars. If it is the case, you could imagine that some of these organisms might have survived hidden in the subsurface if there is liquid water available and also redox potential. I'm not sure if it is the case, but it is a possibility, and so potential environments to study on Earth are the subsurface and also evaporites looking for potential biosignatures. So this but essentially you can think on perhaps exoplanets and things that are more distant in other solar systems. And with that, I finish. I'm ending here, and I thank you for your attention. Thank you.